So the next presentation will be by Benjamin Winkeljann. He comes also from Munich as Leon and a little bit in the same area. It's about inhalable RNA. And I learned from Benjamin just this morning that he's also mechanical engineer. So I just want to say to our mechanical engineers, so there's always a second career possible. You can even move further into pharma, but also for you, Benjamin, I mean, we always need good mechanical engineers.
Yeah, thank you very much for the intro and also, of course, for the invitation. I mean, it's really a pleasure for me to be here and to present today our technology on inhalable RNA therapeutics. And yeah, if you were watching carefully during the factory tour, you may have already learned quite a few aspects about inhaled products. And lucky for me, we just had a presentation from Leon before, so another aspect that I would have to introduce, the so-called lipid nanoparticles, have already been covered, and I can actually jump in right into my presentation and into the material. Yeah, as I said, and you may have guessed, so RNhale, the name in the end says it all. So we are working on inhaled RNA therapeutics. And what differentiates us from most other people that are working on inhaled RNA therapeutics at the moment is that we have developed a technology to manufacture these RNA therapeutics as dry powder. And that's substantially different, I will come to that later, to current approaches that are mainly used on fluid nebulization. Yeah, we are located in Munich, as Marco has already introduced us. We are technologists, so not the classical biotech path that comes from target identification, drug molecule, lead identification through the entire process into the clinic, but we came from the technological perspective, meaning we have developed a technology that facilitates certain roles that I will talk about, and from that point, we have identified for what indications, what therapeutic use that therapeutic technology could be useful. At the moment, we are developing also our own clinical candidate. We have been generously funded by the European Union recently to conduct the IND-enabling studies for a therapeutic of severe asthma. And that should serve as kind of a forerunner project also to establish the technology I'm going to present to you as a platform for inhaled RNA therapeutics. But let's start one step earlier. I mean, we have heard about why RNA therapeutics are interesting. So there's a lot of potential to, in the end, always achieve the primary goal of correcting a protein expression in the human body. That can be there is too much of a protein that's causing inflammation, you want to downregulate it. That can be there is a misfolded or wrong protein expressed, so you want to correct the DNA. But that can also be that you want to produce a protein that has not been there before, like for example in the COVID vaccines, where you want to express the spike protein to induce immune recognition of the body. But the end is always you want to correct a protein, and that can be interesting for various diseases. For us, primarily focusing on lung diseases. But now if you think about, you have a big complex molecule that's additionally very sensitive, like RNA. It's by far not trivial to bring that into the human body. And these are mostly technological challenges that have to be overcome. So if I would like to summarize, of course, if I want to bring a molecule into the lung by inhalation, the first, and that sounds trivial, aspect is that your drug product is inhalable. Inhalable means mostly physical characteristics, so it has to be in a state that it can be aerosolized, that you can inhale it actively. But also, of course, considerations such as particle size, where it deposits, and so on. I will come to that in a few seconds. Second aspect, and that's particularly important for the lipid nanoparticles that have been introduced before, is that lipid nanoparticles, RNA-loaded lipid nanoparticles, they are particularly sensitive to mechanical and also thermal forces. Mechanical forces, those of you who have been a bit informed about what happened during the COVID vaccines, like if the medical doctors got their vials with the vaccine shots, in the introduction for the medical doctors was written, do not shake it. Because the product is that sensitive that excessive shaking would create enough shear force for the lipid nanoparticles to partially disintegrate, lose their cargo, lose efficiency, and so on. And now, if you imagine one step further, you want to aerosolize a solution containing lipid nanoparticles, as I try to illustrate here, of course you're generating massive shear forces during the droplet generation. And that's a particular problem if you want to administer using liquid aerosols. And then, addition on all those mechanical, physical aspects that I just introduced, you have the biological barriers. I mean, also when you consider the lung, it's not like, okay, you brought it into the lung, it's at the target location. No, it's by far not. It has to cross several biological barriers, starting from this slimy substance, this mucus that you find all over your lung, that's in lung disease patients usually even more pronounced than in healthy patients, over the cellular uptake to get into the correct target cells depending on which indication you want to target. So you have a lot of factors that in the end determine whether you can achieve a successful delivery of RNA into lung cells. And that's why at the moment many clinical trials that are out there that are mostly using these nebulized fluids are still suffering from insufficient delivery of the RNA to the envisioned targets. What our solution is, and now I try to illustrate it, but it requires a few steps that we have to think through what we are actually doing here. So what you see here on the left side, just to introduce that first to you, this is our powder that's actually being blown into an aerosol. That was during one of the mouse studies that we conducted, where we used this insufflator device to make the mouse inhale our dry powder product. But what actually do you see in here? So if you would zoom into this powder, you would find tiny little particles in the size of ideally 1 to 5 micrometers. And that's what I tried to illustrate in the schematic here. So this large sphere that you see, this is the dry powder particle that is in the size of what I just described, 1 to 5 micrometers. And what we do is, into this dry powder particle that's composed of a stabilizing excipients, we embed those little lipid nanoparticles that contain the RNA. And now this like two-layer system, essentially, it comprises 2 key features that we need to achieve. We have talked about the lipid nanoparticles and their role in RNA delivery, so protection and the ability to cross biological barriers and transfect the cells. What the excipient matrix does is essentially capture and stabilize these lipid nanoparticles in a dry form where they are protected from shear forces, from temperature, and where we are facilitating a physical form that allows for creating a dry powder dispersion and therefore for facilitating inhalation. So yeah, here is a little video of actually, just to show you again, this is really dispersible. It's a powder. That's what the patient in the end is supposed to inhale and bring into their lungs. And my micro is moving a little bit. To step a little bit into how the manufacturing process for that works is, so we are starting here on your left side. So what we have here is is the nanoparticle assembly process that we have just learned from Leon before. So imagine here is the Leon device. We have our target RNA. So we are mostly working with siRNA, but I don't want to go too much into detail on different RNA modalities in here. We have our lipids and we formulate lipid nanoparticles. So that's identical to the process that we have seen before. But what we do subsequently is to blend that with an excipient and then feed it into a spray dryer. So that's the device that's illustrated here. Spray dryer essentially works by dispersing a solution that contains here lipid nanoparticles plus excipients into a drying chamber. And while these little droplets are traveling through the drying chamber, the water evaporates because the chamber is heated, leaving these microparticles that we have just seen before on the exit. And what then comes afterwards, so out of the spray dryer, we have the powder with the correct characteristics to be filled in an inhaler. And that's then the part where Haro will come in, because this powder has to be loaded into capsules or blisters to be administered in the patient later on during the fill and finishing process. So that's the process to get to inhalable dry powders. So now if we are thinking about what requirements does this powder have to fulfill in order to meet the expectations that you have for such a drug product. So one, of course, is you need to have a sufficiently high API concentration. That's in our case the RNA, so the active ingredient. And I am highlighting this here so you could think, yeah, okay, just put more lipid nanoparticles in there or use more powder, but you have a certain framework for this. So on the one side, you don't want to let the patient inhale grams of powder, because at one point it just feels like swallowing sand. So there is a natural limit of what the patient can actually inhale. And then the second part is the ratio between excipient and API determines a bit the stability, let's say, of the RNA. The more excipient you have, the better it is protected, for example, from thermal forces during drying, but also later on processes during storage and redispersion again. So there is a certain limit. That's one requirement. API activity, of course, that was one of the technological key challenges that we had. How can we dry this RNA-loaded lipid nanoparticles without losing RNA integrity? Because, as I said, RNA sensitive, that's by far not trivial. And the third is the inhaled properties that we had, so these 1 to 5 micrometers that we envision as a target size for our microparticle product in order to be inhalable and to reach the deep lungs of the patients. These 1 to 5 microns, this is not something new. That has been established and identified with the first inhaled products that have been developed, and it essentially builds on a model that says what considerations do you have for a particle that's being aerosolized and flies into the lung in order to reach the deep compartments. Imagine a particle that's fairly large. If you inhale that, it will not be able to follow geometry of the lung. So everything that's too large will impact in your upper throat. Eventually you will swallow it, it will be degraded in your stomach, it's not useful. So that happens with too large particles. Too small particles also have a problem. If they are too small, they will be able to follow the geometry of the lung, but they will not sediment. At least not if you don't hold your breath for 10 minutes or something like that. So there is a certain sweet spot that had been identified before, and what you can see here is essentially how particle aerodynamic diameter, so we're not talking about geometric diameter, but aerodynamic, that considers also the density of the particle, how that affects deposition behavior in the different regions of the lung. So this is an important characteristic also when you think about your patient and the disease that you're targeting. Is the disease more located, for example, asthma that's generally located all over the lung, or do you really need to reach the deep lung? So there's certain characteristics on the dry powder that you also need to consider. Now I've been talking about excipients a lot, and I guess some of you at least were wondering, what excipients can actually be used there. And for our purpose, we decided to go a bit of a de-risk approach here, because we have a complex molecule, complex disease targets. We have a new manufacturing process, so we don't want to bring in another variable with excipients that have not been used for inhalation before. So we were essentially looking at, okay, what excipients are out there that have already been used for inhaled products and have reached at least a certain level on the development status, so that we can expect to get clinical clearance for that. And these are all the excipients that are out there. It looks actually fairly a lot, but if you take out all the buffer salts, it pretty much reduces to a few left ones, where there are still materials also that are more used for capsules rather than for the inhaled product itself. And here a few, those are highlighted in red. Those are excipients that have not yet been approved in an inhaled product, but at least have been far advanced in the clinic and are expected in the inhalation community to be approved in the next couple of years. So starting from that, we decided, okay, let's go into a formulation screening. Let's see what excipient or excipient combination fulfills the role that we expect for our dry powder product. And starting from that, we decided to go with 3 base excipients. So lactose on your very left, trehalose in the very middle, and mannitol as a crystalline excipient on the very right. And what you already see here in the first line in these SEM images is that when using lactose or trehalose, you really get nicely spherical particles, like almost like how I illustrated them in the sketch before. And you also see the size bar, so fairly below 10, even the most of the population below 5 micrometer. So actually in the target range that we are looking at. If you're using mannitol, totally different image. So if you, as a background information for that, if you're spray drying pure mannitol, you get perfectly spherical particles. But by adding the lipid nanoparticles as a component in that system, you dramatically change the physical properties of the powder, and you get something that looks far away from being inhalable at all. Also, as a second step, we were looking at the physical properties of the powder. So we were performing X-ray diffraction measurements, and what you see both for lactose and for trehalose, independent actually of the lipid nanoparticle content that we have in there, you see this typical halo structure. You don't see any prominent peaks, so no indication of a crystalline structure in the matrix. Whereas if you look at the mannitol, you really see the different peaks here that indicate a highly crystalline internal structure of your excipients. That's important to consider also, because whether you choose a amorphous or a crystalline excipient determines also stability of your product, determines tolerability of the patient, for example, also. So for example, mannitol is something as a crystalline excipient that shouldn't be really used if you have inflammatory processes, for example, as in asthma going on, because it can also further trigger inflammatory reactions. And also solubility. I mean, the idea is at one point when this particle impacts the matrix excipient has to disintegrate again, because it has to liberate the lipid nanoparticles in order for them to transfect the cells. So also considering these aspects, we are more prone to go towards amorphous excipients than to crystalline excipients. But lucky for us, I mean, those are also the one which initially with the base excipients showed already the more promising physical properties in here. What was also important for us, of course, is to assess whether the excipient plays a role in preservation of the RNA activity post-processing. And one of the early kind of high throughput readouts that we performed in there was to investigate the performance of our RNA. So here shown as a knockdown performance. That means we are knocking out certain genes that are disease causing in a reporter model. Reporter model means we're using cell lines that are stably expressing fluorescent proteins. So if you're knocking down these fluorescent protein, you will see a decrease in fluorescence intensity that you can use then to quantify activity also. And that's what you actually see in here. So you see GFP expression of a untreated cell sample versus fresh nanoparticles, spray-dried nanoparticles in the different excipients. But what you actually see here is that although the physical state of the dry powder looked dramatically different between the different formulations, the activity, at least in this initial screening assay, does not indicate any RNA damage or loss of potency in that regard. But now there comes another factor into play, and that is the inhaler. Because if the inhaler and the powder doesn't match, you will not get a sufficient dose into the lungs of the patient. So what we did here is we used two different inhalers. There is a manuscript in preparation, so my colleague Leonie is also here today. So she actually conducted and led most of these studies that will be published also later that year. At the moment I cannot disclose what inhalers we have been using here, but just imagine two off-the-shelf dry powder inhalers with different, let's say, dispersion mechanism that we used here. And what is important here, you see dramatic differences between the two different inhalers, and also between the different excipients that we have been using. So that actually reflects, like, can the powder in combination with the inhaler be nicely aerosolized, yes or no? And here you see the fine particle fraction, which is essentially the part that we will later end up in the lung, that dramatically decreases when we use mannitol here instead of trehalose and lactose. And also between trehalose and lactose, you see that the performance of the second inhaler is much better for the lactose. So it seems to be more forgiving towards the dispersion mechanism than does the trehalose. That was up to now the initial screening using our base excipients. We extended that. I will not show you all the data, just to show you what different excipients combinations we use. So we blended in leucine into our base excipients. Leucine is quite interesting because it's a hydrophobic excipient. It's being used to protect powder also from sucking up too much moisture. So powders, if they become too hygroscopic, that's disadvantages, especially for storing, but also during fill and finish, because they can clump together, then they will not be dispersed nicely anymore. So we blended in leucine as different concentrations, and then went through a big set of parameters. So we looked at production parameters, classic ones like yield. If you just recover in the end 40% of your drug product, it's not commercially viable commercially viable process. We looked at EGFP knockdown, as I said, recovery of RNA, but also the different inhaler concentrations. And in the end, we found out that we basically have the best performance when combining lactose with the different leucine concentrations. There are further studies ongoing also on the hydroscopicity and on the storage stability. What we have shown up to now is that we can store our drug product for 18 months at room temperature. This is at least 18 months because then we run out of samples, so we couldn't continue the study. But if you compare that, talking back to the COVID vaccines or to other RNA therapeutics, where you always have to consider during storage, but also during logistics, these low temperature cold chains. I mean, this is quite some advantage that comes on top of the fact that this is an inhaled product, but it also comes with the advantage of global accessibility of drugs, distribution chains, and so on. But now, what can we do with it? I was talking about powder. Now I want to talk a little bit, at least, about our indications that we are addressing with that. So at the moment, we are mostly interested in the first three of here. So our lead candidate is against severe asthma. I will come to that in particular afterwards. But we are also working in two cases with collaborators, both on therapeutics for COPD, where we have a really big unmet medical need, but also on the development of antiviral therapeutics. Because RNA is also particularly interesting as a quick response tool for developing RNA, newly developing RNA viruses, because you can rather quickly adapt the RNA sequence without having to change anything else in your process, and thus being faster able to respond. So, but now we will talk a little bit about severe asthma. And to understand what is the medical gap here that we are addressing, we first have to understand a little bit how the patient population of asthma looks like. I mean, statistically, we probably have a few asthma patients here in the room, but even if we don't have, you for sure know someone who has been using an asthma inhaler before. So it's quite a common disease, especially in the developed countries where also allergies are very present. But if you look into asthma, you actually have to first classify which asthma severity we are talking about. And for us, of course, a mild asthma patient is not the one who will later receive expensive RNA therapeutic. The target population for us is really the severe and uncontrolled asthmatic disease that is being addressed with our therapeutic here. And the second differentiation that you have to make is if you're targeting T2 or T1, or in other words, eosinophilic and non-eosinophilic asthma. At the moment, there is only one drug out on the market that targets T1 asthma in its severe form, and T1 asthma makes up to 40% of the entire asthma population. So that's a substantial subset of these patients also. If we now look one step further, so I said there are drugs available. These are essentially all the drugs that are currently out there for the therapy of severe asthma. All of those are antibody-based drugs that are systemically administered. And that comes with another problem. So antibody-based drugs are very prone to induce the formation of so-called anti-drug antibodies, meaning the body reacts to the drug in a way that it renders it ineffective. And that's a problem that comes up depending on the drug being used in 5 to 10% of these people receiving these antibody-based drugs, and those do not have a therapy at all. What physicians then typically do is they exchange one or two of these drugs, say, okay, if Dupixent doesn't work, I'm using Tezspire. But if you're already prone to develop anti-drug antibodies towards these kind of medications, the chances that you will find a suitable technology there are generally rather low. And that's also something that we can address with our technology, because it's not, first of all, it's not acting extracellularly, and that means it will be in the cell. It will not permanently circulate like the antibodies that we have here, and thus it's being less prone to also being rejected by the body or neutralized by these ADAs. Yeah, our therapeutic target is actually, if you look at the disease indication, so in asthma we have certain symptoms in the end that causes the disease as we know it. And if we look then back what molecular markers cause these diseases, we can go further upstream, and eventually we will land at the protein TSLP. That's an alarmin, and that's one of the first most upstream proteins that involves or is involved in the progression of asthmatic disease and its symptoms. So what we are doing with our technology is essentially we are inhibiting TSLP very early on, and by thus reducing the symptoms and the disease progression in these severe asthma patients. What you can see on the left side is actually that this is a clinically validated target, so that's not something that we draw out of our pockets, but that has been shown in clinical patients that this is really strongly connected to the asthmatic disease. We have run 2 important studies on that as a proof of concept for our technology. One that was conducted in so-called human precision cut lung slices. So that's a model, essentially a mini lung model of human patients. So we receive patient material from the clinic, from biopsies that we can cultivate in our laboratories, and we can perform certain studies on there. And what we essentially did here is that we wanted to look at the downregulation of the TSLP gene expression, so our target gene, if we can get that down after it has been triggered by a response, and also, of course, the TSLP protein expression as a more important readout. And what you essentially can see here is that we have downregulation to base level, essentially in both cases, really highlighting the potency also of our technology. Importantly, also, of course, we are looking at inflammation markers, because most inhaled therapeutics do not fail because of a lack of efficiency, but because of a lack of safety. So safety was also always one of our core aspects that we were looking into, and you essentially see that the relevant cytokines, I mean, we run large panels there, but the relevant cytokines do all stay at a normal level, even if we go there in with rather high doses. Complementary to that, we run an in vivo proof of concept. So asthmatic, oh, that's a bit unsharp, sorry, that's ugly. Essentially an asthmatic mouse model where we administered our powder. You remember one of the early short videos that I showed, so really the powder, not a redispersed product or anything else. And what we essentially saw in that mouse model, and that's an important readout here, is that the diseased mouse really quickly develops this perivascular inflammation. So you see here this high inflammation around the vascular regions, and that we can really effectively reduce these inflammation symptoms with our treatment, like even in that short-term model. Additionally, we were here looking also at these cytokines. So these are the downstream cytokines that I showed you before. So the very last step that induces the actual symptoms, and also all of those are downregulated, 2 important ones, even down to base level, while the safety profile looks really perfectly fine. Yeah. I don't want to summarize that again, so I give you an abrupt ending, but I'm happy to answer additional questions if you have interest into that.